Hello everyone, let me introduce my poster titled Genomic Features of Multidrug Resistant Beijing M Tuberculosis Clinical Isolates from Kazakhstan. According to WHO, tuberculosis is the 13th leading cause of death and the second leading infectious killer in the world. Kazakhstan is one of the 30 countries with high rates of multidrug resistant tuberculosis, MDR-TB, the severe form of TB that is associated with the resistance to the main anti-TB drugs, isoniazid and rifampicin. Different studies revealed the association of Beijing genotype with antibiotic resistance and the massive distribution of MDR-TB. The aim of this study is to identify the genomic features of MDR and TB isolates by genotyping methods and whole genome sequencing for the first time in Kazakhstan. In our study, 200 MTB clinical isolates from different oblasts of Kazakhstan were gathered from new and recurrent TB cases. All the isolates were genotyped by mirror VNTR methods. And afterwards, 8 MDR-TB samples with interesting clinical cases were chosen for whole genome sequencing. And these 8 MDR-TB samples were additionally genotyped by 2 other methods, polytyping and IC6110 RFP typing analysis. Whole genome sequencing was performed using Roche next generation sequencing platform, and variety of programs such as FastQC, Patrick, in-house prepared R-script, etc. Were used for genome analysis and variant calling. 3 databases and 2 other tools were used for identification of gene mutations associated with drug resistance to the first and second line antibiotics. Comparative phylogenetic analysis was done by phylogeny tool called CSI. And the results of mirror VNTR genotyping of all isolates showed that Beijing family is the most spread MTB family among all isolates, and they were found in 57 and 93% cases among new and recurrent TB groups. Among 8 MDR-TB isolates subjected to WGS, 75% isolates belong to Beijing cluster 9432, which is prevalent in former USSR countries, and the rest 25 isolates identified as Beijing cluster 132 that prevails in China and Russia. All MDR-TB isolates had a Beijing-specific polytype, and all isolates with Beijing's polytype were placed in modern lineage 2, East Asian lineage. It can be seen in here. Whole genome mapping and statistics are shown in table 1, and the whole genome sequencing showed several new genetic variants in our in drug resistance genes in our study. Drug resistant profile of all MDR-TB isolates are shown in table 2, and all mutations found in genes associated with resistance to the first and second line antibiotics can be seen in here. Just would like to note that the interesting finding in our study was that all isolates, 8 isolates, had the same GRA mutations seen in all fluoroquinolone resistant susceptible strains. This is the second line antibiotic, but one isolate had a mutation, additional mutation in GIR-B gene, which is known to confer resistance to FOQ, the second line antibiotic. Phylogenetic analysis showed that isolates grouped into 2 clusters. The first cluster consisted of 7 MDR-TB isolates, was closely related to B0W148 strain sequenced in Russia, and the last clinical isolates from another obtained cluster is phylogenetically was similar to the strain that was isolated in India. And in conclusion, Beijing genotype prevailed among both new and recurrent cases in our study, probably because Beijing genotype is associated with drug resistance and chronic forms of TB. As I mentioned above, Asian Russian cluster was the largest and dominant subtype in Kazakhstan among MDR-TB isolates. All MDR-TB samples were placed in modern lineage 2, and whole genome sequencing of MDR-TB isolates was done for the first time in Kazakhstan, and the results showed that most of isolates was closely related to B0W148 isolate that was sequenced in Russia. Potential new genetic determinants of drug resistance were identified, and understanding the complex biology of TB drug resistance and the pathogenesis of the disease will provide new knowledge for the development of improved diagnostics, vaccines, and new anti-TB drugs. And the genomes of Kazakhstani MDR Beijing isolates were uploaded to the NCBI, and can be used for comparative genomic analysis with the genomes of isolates circulating in other countries of the world. Thank you for your attention.